Hello everyone, my name is Sagar Sridhara and I'm a postdoc in Institute of Molecular Biophysics in Florida State University. The title of my short talk is Structural and Functional Basis of Biomolecular Assemblies. I would like to give you a brief overview of the projects that I'm currently working on and also the projects that I have previously worked on. So currently I'm working on one of the types of CRISPR-Cas systems called the type 3 CRISPR-Cas system, wherein it's a characteristic feature of bacterial immunity. The bacteriophages are viruses that invade the bacterial cell and utilize the host transcription translation system to propagate itself. The end result of the phage infection is the death of the bacterial cell. However, bacteria has its own defense mechanism through which it defends itself from the phage infection. That is achieved through CRISPR-Cas ribonucleoprotein complexes that specifically bind to the phage RNA transcript and acts as an RNase to degrade the viral RNA. To achieve this, the host genome encodes a set of Cas genes and CRISPR-Cas and a CRISPR array. And this CRISPR array basically contains several short spacer sequences that are arranged in between repeat sequences. It is these unique spacer sequences that are key to identify the invading RNA transcript. So in the event of the injection of the viral DNA into the bacterial cell, the host CRISPR-Cas RNP or the ribonucleoprotein complex expressed from its genome would identify and bind to the CRISPR RNA present within the effector complex. The binding event would trigger a cascade of events that would cause certain conformational changes in the ribonucleoprotein biomolecular complex that would activate its RNase function to degrade the viral RNA. So this is what I am showing over in the second part of the slide. Moreover, this effector complex would bind to another unique protein to get activated as a DNase and targets the viral DNA. Thus, the ribonucleoprotein would have successfully degraded the viral RNA transcript and the viral DNA, leading to the survival of the bacteria. So this is just an overview of the CRISPR-Cas system. The aims of my current project is to structurally elucidate the type 3 CRISPR-Cas complex intermediates, wherein we determine the structural basis of events that lead to viral nucleic acid degradation. We also aim to understand the cascade of transition events of the type 3 CRISPR-Cas complex. Specifically, we want to understand as to how the ribonucleoprotein switches from an inactive state to an active state to function as an RNase, and also understand how this ribonucleoprotein could function as a DNase. To achieve this goal, we use a combination of biochemical enzymatic assays and structural biology tools, mainly the cryo-EM and of course crystallization. In this slide, I'm showing one of the chromatogram, size exclusion chromatogram of the formation of the CRISPR-Cas complex in vitro. Specifically, the type 3 CRISPR-Cas system is composed of 5 protein subunits and 1 CRISPR RNA, which acts as an effector complex of unknown stoichiometry of individual subunits. Here, the individual subunits and the CRISPR RNA are expressed and purified separately. And as you can see, the complex is formed on a size exclusion column. The chromatogram basically separates out several unique species of protein-protein complexes and also protein-RNA complexes, which are all of significant interest to us. We would subject some of these species that are above, say, 150 kilodaltons to cryo-EM, and while several subcomplexes, which are basically coming in the later part of the chromatogram, are subjected to crystallization experiments. If we are able to obtain crystal structures of these subcomplexes, they will, for example, facilitate the model building of electron density maps generated by the cryo-EM of the full complex. So, here I would like to end the current project overview over here, and I will proceed to the next slide, wherein I'm talking about my the project that I've previously worked on. So this is completely different from what I've just spoken. Here, I would like to just highlight one of my previous projects that I was involved as a PhD student in Karolinska Institute in Stockholm. The aim of this project was to understand the structural and functional basis of human mitochondrial tRNA processing. So in this slide, you can see a schematic representation of transcription within the human mitochondria. The human mitochondrial matrix contains several copies of mitochondrial DNA, and this mitochondrial DNA is short, circular, double-stranded molecule of around 16.5 kilobases. The mitochondrial DNA encodes information of 13 essential protein subunits involved in oxidative phosphorylation, along with 2 ribosomal RNA elements and 22 transfer RNA elements. And the transfer RNA elements punctuate most of the mRNA and rRNA elements. The transcription of the mitochondrial DNA is performed by mitochondrial RNA polymerase, which is abbreviated here as POLRMT. So that would generate long polycystonic precursor RNA sequences. So basically, the transcription of 2 strands of mitochondrial DNA would give rise to 2 polycystonic precursor RNAs, and this scheme is basically showing one such polycystonic RNA. In this scheme, only one is shown, and the long RNA transcript must undergo 2 key processing events to liberate individual ribosomal RNA, individual transfer RNA, and individual mRNA elements. The 2 key events are basically the 5' tRNA processing by the mitochondrial RNase-P and the 3' tRNA processing by the mitochondrial RNase-C. So as the scheme pretty nicely shows here, 3 proteins called MRPP1, 2, and 3 are acting as a mitochondrial RNase-P, while the other 3 proteins, MRPP1, 2, and ELAC2, acts as a mitochondrial tRNase-Z. So the end result of these 2 events is basically that all of the individual RNA species can undergo their own set of maturation. And this is mediated by a lot of unique enzymes, and we are not going to go into details of each of them. But I would just like to highlight one such, one of the sub-projects that I was basically involved in, wherein we were trying to understand the crystal structure of MRPP3 protein, among many others. So we were trying to crystallize the full-length MRPP3 protein, but unfortunately we were not able to generate any crystals of the full-length MRPP3 protein. What we did was to generate a series of truncated variants of MRPP3 protein. So here you see a schematic representation of MRPP3 protein. It has a mitochondrial target sequence which helps the protein to make its way into the mitochondria. And it has 3 domains. The first domain is the N-terminal pentatrichopeptide repeat containing domain. It has a central domain, and it is followed by a C-terminal metallonuclease domain. So when we did not get the crystals of the full-length MRPP3 protein, we generated several truncated variants by successive deletions of 1 or more PPR repeats. So one such variant, which started from residue number 207 until the residue number 583 crystallized, and we were able to basically screen all these different variants for crystallization. And finally, we got a very high resolution crystal structure for of the variant which started from residue 207. Thus, the optimization of crystallization and optimization of crystal hits have only positively contributed to my projects. And this is where I would like to end my talk. Thank you.